Yesterday, we heard a lot about risk assessments, and today, during the morning also, and in the presentation of Koen, we had some nice examples of case studies of extractable studies. And this one is, let's say, the next step in the sequence is on leachables, and also really focused on practical case studies. It may seem a little bit redundant, but in the introduction of the presentation, I would like to come back to the definition of what a leachable actually is. And then the core of the presentation are the case studies. And then at the end, of course, we will come back with some conclusions. So what is a leachable? I think everyone in the room knows what a leachable is. But I really wanted to look for a formal definition, and I found one in USP 1664. But there it is mentioned in the context of a packaging system. And we need to consider, if you have the final product in the container, there are leachables coming indeed from the packaging system. But on top of that, you have the leachables from the production materials, which are often referred to as pearls, process equipment related leachables. I think that is the terminology which is used in USP and also in some other of the guidance documents. With respect to regulatory documents, I just wanted to give an overview and compare the publication dates of some of the documents on container closure systems compared with the one on process materials. So the initial concern about extractable leachables was on container closure systems because there the risk is higher. And at a later stage, there was also a lot of interest and concern with respect to the process materials. So the documents that you now see on the slide are the first documents, and which say that you need to address extractables and leachables. And then later on, a series of documents was published, in which more practical aspects were discussed, which analytical program that you need to use, how to establish extraction procedures, and so on. What is an AET? What is a safety concern threshold, and so on. So a very relevant document, I would say, is the one from PQRI from 2006, which is on orally inhaled and nasal drug products. So that document really contains or gives you some guidance about how to perform extractable testing, which analytical techniques you need to use, and so on. And then later on, also some similar documents were published. There were some white papers coming from BPSA. And also some papers from Bioforum or BPOC. And then there were the USP chapters, the ones for container closure systems and delivery systems, were 1663 and 1664. And those final versions were published in 2020, while the USP 665 and 1665 chapters on process materials were finally published 2 years later. And also, if you would look to the draft dates of those documents, I think the first draft of 1663 and 1664 was published in 2014. For the process material USP chapters, it was 2017, I guess. So there's always a couple of years in between those publication dates. So leachable studies, when do we need to perform those actual analytical studies? Well, in the context of a container closure system, for us it's almost a standard assumption if a customer comes with a request to perform an extractable study on a container closure system, that we will do a leachable study also afterwards. We even quite often provide them immediately with a quotation also on the leachable study, because often it's it's already good that you have an idea about the budget in advance. And this table that I show here on the slide was actually originally published in the FDA guidance document of 1999. And it does not really say when you need to perform leachable studies. It just describes the degree of concern associated with the route of administration and also the likelihood of interaction between the dosage form and the plastic. So nowadays, I would say that in almost all cases for a container closure system, you would need to do extractable and leachable testing. Maybe the only exception would be the right corner below in the table, the oral tablets and oral capsules. There you could get away with some limited extractable testing and compendial testing. For process materials, it's, as we know, different. And the main reason for that is visually represented here on the slide. There's a greater distance between the actual process material and the patient. So leachable studies are definitely not a requirement for all process materials because the whole goal of risk assessment is actually to omit high-level extractable testing and leachable testing. So in which cases do we need to do leachable testing? What do the recommendation and guidance documents say? If you look to the BPSA white paper of 2010, the Bioforum document and the USP chapter, then they say, and we start from the assumption that we need to do high-level extractable testing. In the context of USP 665, that would mean we need to apply the standard extraction protocol. Okay, we do the extractable study, and then we look if we find or detect compounds above the final AET. If we do, then we need to do a tox assessment. And then what we actually do is compare the exposures of the compounds based on the concentration in which we have detected them in the extractable study. We compare those exposures with the permitted daily exposure, which is a compound-specific toxicological threshold. If there are compounds which are present above the PDE, then we need to do a leachable study, because at that point, that is the only way or strategy to show risk control over our process. So apart from that case, are there additional scenarios in which leachable studies should be performed? Yesterday that was also discussed. Guidelines and recommendation documents, they do not really say in a hard-coded way what the authorities actually expect. And there's always some room for interpretation. And sometimes for some aspects, the authorities could have some more stringent requirements than what is actually written in those recommendation documents. So what we try to do as best as possible is collect direct and indirect feedback from our customers, and based on that also give the right recommendations back to our customers in which cases they actually need to do the leachable study. So apart from the case mentioned on the previous slide, so if we have extractable compounds present above the PDE, we know that the authorities often expect that leachable data are generated on filling line process materials. From a scientific point of view, it might make sense because there's direct contact with the drug product, and immediately after the contact, the container closure system is filled. And then the other type or the other case in which a leachable study may be necessary is when you're dealing with a storage application, a storage bottle, a storage bag. I was actually glad to hear yesterday from the presentation of Marine and Etienne that they also confirmed that authorities often expect leachable data on process materials which are used for storage. That is a good thing because then this conclusion or this statement is not too speculative. So yeah, I would say from a scientific point of view, it might also make sense because often leachables are formed because of the effect of time, and that is often not covered in an extractable study. So an antioxidant, for example, could degrade over time. That degradation takes several weeks or months, and you will only detect that compound as a leachable and not as an extractable. And such a degradation compound could have also different toxicological properties as the actual antioxidant. So in such a scenario, it definitely makes sense to do a leachable study on process materials used for storage. Now going to the case studies. So the first case study here is a relatively simple one and also very comparable with a leachable study on a container closure system. It's one on a storage bottle, so it's practically very similar. And later on we will discuss some more diverse case studies. So this case was a 5-liter PET bottle with a polyethylene cap and used for the storage of a drug substance. Yeah, I need to be a little bit vague about the composition of the drug substance, of course, because of confidentiality reasons. The contact time and temperature were 12 months at 5 degrees C. What is our motivation here to do a leachable study? It is a storage application, so you have the effect of time. It is expected by the authorities. That is our idea based on feedback. And we also had one extractable present above the final AET in the extractable studies. So the study setup, well, the study was actually performed on a small scale bottle because the 5-liter bottles were a little bit too large to fit into our incubators, and that would also consume a lot of drug substance for our customers, of course. We stored the bottle in inverted conditions, that there was also worst case contact with the closure. And we stored it at 5 degrees C. We took time points we took samples at T0 and after 1 year. And the analytical techniques that we've included were headspace GCMS, GCMS, and LCMS to screen for our organic compounds. And then on top of that is quite standard. We include a method suitability test in our leachable study. That means that we want to prove that we can detect our target compounds that we have seen in the extractable study in the environment of, in this case, our drug substance with our analytical techniques at a relevant level, which is typically based on the AET. So the AET calculation in this case, we relied on the safety concern threshold not on the 1.5 micrograms per day, but on 5 micrograms per day, because this is a non-chronicle application, and then you can rely on a higher threshold. We used a small scale bottle, so we needed to do some normalization for the surface area, and we expressed the AET here in micrograms per square meter. So the method suitability test results then. So we spiked our one target compound, which we have found in our extractable data, in our blank drug substance, which was not contacted then with the storage bottle. And we were able to detect that compound, which is, by the way, a degradation product of an antioxidant, in the complex environment of our drug substance. And then looking to the contact sample, we did not detect any compounds above the final AET. For GCMS, we were able to detect the same compound also with GCMS. There's often an overlapping zone between the different analytical techniques. So we were able to detect it. So we have proven that our methods are suitable to detect the target compound with our analytical methods. And then the GCMS contact results. we also did not observe any compounds present above the final AET. And LCMS also resulted in no compounds present above the final AET, meaning that we can conclude by doing this leachable study that there is risk control over the process if we are speaking about a storage bottle. Then the second case was on the downstream part of a production line. And the leachable study was actually combined with the leachable study on the container closure system. So often price could be an issue to perform, for example, an additional leachable study on the filling line, but we can easily combine it in one study in combination with the leachable study on the container closure system. So we basically do some small adjustments in our study design. We analyze only one contact sample and we can capture both leachables coming from the filling line process materials as well as from the container closure. A big difference here is that the contact procedure and the sampling was performed by our customer. And that is a big difference with, let's say, a traditional leachable study on a container closure system, because there we often do the aging at Nelson Labs in a climate chamber. We take the sample. For IV bags, most often the customer does the aging, but they will send the filled bags to Nelson Labs and we open the bags and do the sampling. But here, the contact procedure and the sampling are under the responsibility of our customer. So looking to the production process, so it was a process used to produce a certain drug product. I don't know which one anymore. I also need to keep it vague in this case. And what was actually in scope with respect to leachables were all materials downstream from the drug substance storage bag. So what we need to do is compare a contact sample with a blank sample, because otherwise we cannot differentiate matrix ingredients and ingredients degradation products from the actual leachable. So we do a comparative analysis between a contact sample and a blank reference sample. So here, in this case, it's a combined leachable study on the container also. So our contact sample is the sample in the container, and the blank sample was sampled right downstream from the drug substance storage bag. The analytical techniques that we included were the 3 organic analytical techniques that we use, and we also included a method suitability test to verify the detectability of the target extractables. What is rather specific to this case study? It is an in-process sampling, and the sampling is by and under the responsibility of the customer, and multiple materials between the storage bag and the final container. There are a lot of materials included, so that also results in a relatively long list of target compounds for which you need to prove that you can detect them in the environment of your drug product. So the AET calculation here, the AET, just take the SCT divided by the maximum daily dose, and then you come up with the AET. You include some uncertainty factors related to the screening character of the methods, and then you come up with the final AET numbers. We only look here to the method suitability test results of GCMS, but they are rather indicative also for the other techniques. And this is a little bit the reality of when you're dealing with a very long list of target compounds, then the chance is quite high that some of those compounds will not be detected in the method suitability test. That is not a problem, and we can easily determine a PDE for those compounds, because the original spiking level is typically based on the SCT of 1.5 micrograms per day. We can then define PDEs for all the compounds that we did not see in the initial method suitability test. PDEs are often a lot higher than the 1.5 micrograms per day value. And then we can do the spiking at a much higher value at which it will be much more likely that we will be able to detect those compounds. And then looking to the contact sample results, for the target compounds, we did an evaluation versus the result in the method suitability test sample, because in the method suitability test sample, we spiked at the critical level. So we want to see how the value in the contact sample compares with the one in the method suitability test sample. For the non-target compounds, we do the evaluation versus the final AET. And what we noticed here, and we also needed to analyze 2 different batches for the sponsor. The batches, there are some minor differences, but in general, I would say that the results are relatively well in line with each other. And then if we look to the evaluation of target compounds, we see in headspace GCMS that there is one compound which was not detected in the method suitability test, but it was detected in the contact sample, meaning that it was definitely present at a critical level in the contact sample, meaning that we need to do some additional work there. For GCMS, most of the compounds were detected below the concentration of the method suitability test sample. Only for 2 compounds, we also needed to do or need to do some additional steps. And for LCMS, the same 2 compounds which were detected in a critical level in GCMS were detected. So again, there we need to address this issue. We need to what we would do is define PDEs for those compounds, spike them again, and see how the new method suitability test value would compare with the value that you detect in the contact sample. And then for the non-target compounds, there was an issue with one compound which was detected above the final AET. So again, in this study, some additional steps need to be taken. The next case is relatively comparable with the previous case study, but here the customer really wanted to do a leachable study on different isolated sections of the compounding and filling line, and also combined the leachable study with the one on the container. Similar as in the previous one, the sampling was also performed by the customer. So this is the overview of the downstream section of the production process for which we wanted to generate a leachable profile. So the contact conditions here were room temperature, and the worst case contact times are 24 hours in the compounding bag, so the blue bag on the left side of the picture, then 4 hours in the transfer tube, and then 6 additional days in the storage bag. So here again, we look for a motivation to do a leachable study. Why would we do it in this case? For multiple materials in the container, we had several extractables present above the final AET. Then it was on request of the sponsor, and there's a storage bag included. There's filling line included in this part of the process. So it is, as far as we know, an expectation by the authorities that needs to be performed. So how does the study setup look like here in this case? We took one contact sample on the downstream side of the storage bag, and we compared that one with a lab-prepared blank sample. Sometimes it's practically not feasible to take a blank or an appropriate blank from the production process. Sometimes sampling could be an issue, and in that case you can make a lab-prepared blank. And we wanted to compare that contact sample with that lab-prepared blank. Which leachables are then in scope? The ones from the compounding bag, the transfer part, and the storage bag. And then the other contact samples that we wanted to take were filled already in the container. So here, on top of the scope of the leachables of the first contact sample, also the leachables of the container were in scope, and we did the analysis of one sample which was stored for 10 months, and then we analyzed 3 batches at the end of shelf life of the drug product, which was after 18 months. Analytical techniques that we used for this case were our organic analytical techniques, and on top of that we included ICP to measure metals, and again we did a method suitability test because of the long list, and again the list was very long because a lot of test items are in scope of this study, each having all a separate list of extractables. So I will not discuss results of this case study because I think the most interesting thing about this case study is the setup of the study. The results are not always that interesting. And this last case study is one also on tubings used for filling. And we did a worst case simulation at Nelson Labs using the drug products. So this tubing was used for a production process of a drug product with a maximum daily dose of 13.5 milliliter. So maximum daily dose, again, that is really the most important information that we need to have to calculate an AET. And at Nelson Labs, we are all, I would say, the scientific experts. They are all ENL geeks, and if you would compare a normal person versus the ENL geek who works at Nelson Labs, if you would ask a person who works at a pharmaceutical company, at which company do you work and which drug do you produce, a normal person would like to know where the drug product is used for. An ENL geek would like to know the maximum daily dose, because calculating the AET is really one of their favorite activities. So the contact conditions during the process are again room temperature with a maximum interruption time of 4 hours. So why would we do a leachable study? We noticed some extractables above the final AET. It was also a request of the sponsor, and again it's filling line, so could be an expectation of the authorities. So what did we do for the study setup? The actual use in the process is that a drug product will go through the tubing, normally transient contact, but our customer communicated to us that we needed to include possibly a 4-hour maximum interruption time, and that is really a critical period because then there's no transient contact. It's static contact between the drug product and the material. And in those circumstances, you have a higher chance with respect to accumulation of your leachables. So what we did was we filled those tubings. So it was one with a large diameter and a small diameter. We filled it using a peristaltic pump. We closed it using inert clamps, and we did a 6-hour static extraction. So worst case for the 4 hours. And we did it at 25 degrees C, which is the upper temperature of the normal temperature range use. And then maybe a question for you guys. What would be the worst case tubing? The small diameter tubing or the large one? Who says the large diameter tubing is the worst case one? Who says that the small diameter would be the worst case? You are smart people in the room here. So it is indeed the tubing with a small diameter, because there the surface area that a certain volume contacts will be higher, and the leachable concentration is in general a function of the surface area to volume ratio.
Can I defend my position? Because in the small volume you may saturate it, the leachables by concentration, rather than the large, if you can reach high amount at the end. At the end, then you give it everything in the bulk. So I think it's not so obvious. But in the case of surface area, you are right. But in the case of amount, Maybe not. Depend on the amount maximum tolerated. Do you agree?
No, not really, because this would. No, we can discuss that maybe during the coffee break or during lunch. Then we have more time. But so yeah, ideally you want actually a tubing with a large diameter. I mean from an ENL perspective, you want a tubing with a large diameter, which is a thin tubing. Then you have per surface area also less material from which leachables can migrate into your drug product. And then you also, in an ideal extractable leachable scenario, you want that you have a very high flow rate through your tubing and also a very short one, because the longer the tubing, the longer the transient contact time with the drug product and the tubing. But okay, that's maybe not realistic in a process. Then the study setup, we used the same set of analytical techniques as in the previous case. We used the method suitability test for all different techniques, and we selected the target compounds based on supplier extractable data. So I don't think that we did the preceding extractable study ourselves. Again, the AET calculation using the maximum daily dose, and then the method suitability test results here. There was one compound for which we could not detect in the drug product, so we need some additional testing there. For GCMS as well as for LCMS, all target compounds could be detected. Then for ICP, same results. We were able to measure all metals in the environment of our drug product. For mercury, which we monitor with a separate ICP technique, we were also able to detect it in the drug product. And then the results of the contact samples, comparing the small tubing with the large tubing, we see a very similar result, slightly higher concentration for the tubing with a small diameter. And we saw some traces of trimethylsilanol, which we were not able to detect in the method suitability test. So again, that is something that we need to address. So as a conclusion, maybe first the conclusion of the separate part of the second part of the presentation. I think the main feature that you have for leachable studies on process materials is that you have a relatively high diversity with respect to practical study designs. So we discussed one of the storage bottle. Then we've discussed two different case studies for which there was in-process sampling by the sponsor. And in the second one, we did a comparison versus a lab-prepared blank. And then the last one was actually a simulation of the real use contact that we did at Nelson Labs. And then maybe also an important conclusion, because this really was about leachables, but you don't need to do a leachable study in all cases for process materials. It's only in a limited amount of scenarios that you would need to do this type of studies. So thank you for your attention. I'm at the end of the presentation.